Welcome to a new video. In this video, I am going to present a topic called ICH guidelines. So these guidelines are mainly classified into quality, safety, efficacy and multidisciplinary. Whereas there are 10 guidelines in the quality. Q1 for stability, Q2 analytical validation, Q3 impurities, Q4 pharmacopoeias, Q5 quality of biotechnological products, Q6 specification, Q7 good manufacturing practice, Q8 pharmaceutical development, Q9 quality risk management, Q10 pharmaceutical quality system. Q1 mainly deals with the stability of the pharmaceutical products. Q2 analytical validation. So during the method of development, the scientist or the formulator create and optimize the various analytical procedures in order to accurately assess the drug properties. This mainly involves selecting the appropriate equipment, operating the parameters, reagents and sample preparation techniques. It also includes accuracy. Accuracy means it determines how close the measured value is to the true value. Then precision measures the consistency and reproducibility of the results. Then specificity ensures that the method only measures the intended analyte. Then linearity examines the relationship between the concentration and the response. Then robustness evaluates the method performance under varying conditions. Then validation data. Scientists collect the data to demonstrate the method's suitability for its intended purpose. This includes assessing its performance, consistency and reliability. Then Q3, it is about impurities. In pharmaceutical drug products, impurities refers to the undesired chemical substances that are present in the active pharmaceutical ingredients, that is APIs. These impurities can arise during the synthesis of the drug substance or during the formulation. While some of the impurities may have no therapeutic value or benefits, instead of that, they may create the risk to the patient's safety. If their value exceeds the certain limits, then their presence can impact the quality, safety and efficacy of the pharmaceutical products. Q4 is about pharmacopoeias. A pharmacopoeia is a book that provides directions for identifying compound medicines. It's typically published by government or the medical or pharmaceutical society. These books contain monographs which describe the specific preparations. Q5 quality of biotechnological products. These products, which often contain proteins or polypeptides as active components, they are sensitive to environmental factors such as temperature change, oxidation, light, ionic content and shear. Stringent storage conditions are necessary to maintain these or their biological activity and to prevent the degradation also. Q6 is about specification. A specification is a set of quality standards proposed by the manufacturer or the formulator and approved by the regulatory authorities. It includes test, analytical procedures and acceptance criteria. Good manufacturing practice, that is Q7. It is a system that ensures that manufactured products such as food, cosmetics and pharmaceutical goods are consistently produced and controlled according to the set of quality standards. Q8 means pharmaceutical development. How can we define this one? Means it refers to the systematic steps that are taken by the research people or the manufacturer or the formulator in order to identify and develop a new chemical or biological substance into a safe and effective product for the patient. It includes various stages from early discovery up to the final regulatory approval. The phases which are involved in the pharmaceutical development are: first one is discovery and development. It includes identification, that is identifying the lead compounds and assessing their potential. Then next one is preclinical research. It is about conducting the laboratory and animal studies to evaluate the safety and efficacy of that drug. Next one is clinical research. testing the drug in human subjects through the clinical trials. Then FDA review, that is regulatory evaluation by US FDA or relevant authority. Then finally safety monitoring, that is post approval surveillance to monitor the adverse effect. Q9, quality risk management. So it is about assessing, controlling, communicating and reviewing the risk which are related to the quality of the pharmaceutical products throughout their life cycle. It also helps to optimize the benefits or the advantages while maintaining the equilibrium with the risk. The key aspects of QRM is assessment, that is identifying the potential quality risk, then control, that is implementing the measures to mitigate the risk, then communication, sharing the risk related information. Then finally Q10, that is pharmaceutical quality system. So is a guideline that outlines an effective quality management system for pharmaceutical industry. So it provides a comprehensive model or a suitable model which is based on the international standards of organization, that is ISO, in regarding the quality concepts. ICH safety guidelines. S1 for carcinogenicity studies, S2 genotoxicity studies, S3 toxicokinetics and pharmacokinetics, S4 toxicity testing, S5 reproductive toxicology, S6 biotechnological product, S7 pharmacology studies, S8 for immunotoxicology studies, S9 non-clinical evaluation for anti-cancer pharmaceutical products, S10 photo safety evaluation. Carcinogenetic studies. So these studies are done to analyze the risk of development of cancer due to usage of particular type of drug. So the study is especially needed if the use of the drug is for longer duration of time. Means the drugs which are used in the longer duration of time, they must be undergo these carcinogenetic studies in order to evaluate or to identify whether the drug is carcinogenic in nature or not. Study of structural activity relationship. So this is done when the structure of the drug molecule suggests a risk of carcinogenic, or you can say the carcinogenic risk. Pre-neoplastic lesions. When there is evidence of pre-neoplastic lesions in the repeated dose toxicity studies. Genotoxicity studies. So these type of studies are done to identify whether the compound or the drug molecule, whether it is capable of damaging the DNA or not. So whether this molecule or the drug, whether it is inducing the genetic damage in the body or not, to confirm this activity of the drug, genotoxicity studies are done. Toxicokinetics and pharmacokinetics. So toxicokinetics can be defined as, so it is focused on understanding how the drug substance enters the body and what happens to it within the body, especially during the toxicological experiments. So the purpose of this test is, that is the toxicokinetics studies is, aim to describe the systemic exposure achieved in the animals, correlating it with the dose levels and the time of course of toxicology studies. Then S4 toxicity testing. The objective of this guideline is to do the testing of drugs for chronic toxicity testing in rodents and in non-rodents as a part of the safety evaluation of the medicinal product. So this test is carried out in the rodents and non-rodents in order to check the toxicity, that is chronic toxicity of the drug molecule. Then S5 reproductive toxicology. So reproductive toxicology studies conducted to assess the potential risk of the pharmaceutical products to both male and female reproductive health. That is, what is the effect of drug molecule on the reproductive system of the male and female. So these studies aim to identify and characterize and quantify the effects of substances on fertility, embryonic development, and postnatal development. Then S6 for biotechnological products. These guidelines provide a basic study for the preclinical safety evaluation of the biotechnology derived pharmaceuticals. So it applies to the products which are derived from characterized cells using various expression systems including bacteria, yeast, insect, plant and mammalian cells. These products may have intended indication for in vivo diagnostic, therapeutic or prophylactic uses. S7 for pharmacological studies. These studies help to analyze the potential risk of the drugs on human physiology. So, how much the drug is going to affect the human physiology, that is studied under pharmacological studies. Safety pharmacology studies plays an important role in non-clinical drug development. These studies analyze the potential undesirable pharmacodynamic effects of the substance on the physiological functions of the body in relation to the exposure within the therapeutic range. Means the drug is analyzed how much it has impact on the physiology of the human body, and this test is done within the therapeutic range or therapeutic dose of that drug. The pharmacological studies involve the effect of the drug on various parts of the body or systems of the body. So, safety pharmacology studies typically include the following key components. So, how it is going to affect the cardiovascular system, that is cardiovascular system evaluation. So, assessments done, or you can say the test is done, or the observation is done on heart rate, blood pressure, and electrocardiogram ranges. These studies help to identify the potential cardiovascular risk associated with that particular drug molecule. So, how it is going to affect the heart rate and how it is going to affect the blood pressure, and what are the changes taking place in the ECG. All those things are done in the effect of that drug on cardiovascular system. So the next is effect of the drug molecule on the central nervous system, or you can say the central nervous system assessment. So evaluates the effects of CNS including the sedation, excitation, the motor coordination. It helps to detect any adverse neurological effects. So what is the impact of the drug on the CNS, that is central nervous system, whether it is causing any sedation, whether it is causing any excitation, and how is the motor coordination, and it also help to detect any neurological effects, adverse effects are there, that is also we can find out in the effect of the drug on central nervous system. So effect of the drug molecule on the respiratory system. So it investigates the respiratory rate, depth and other parameters. That is, what is the effect of the drug molecule on the respiratory rate, that is on inspiration and expiration, and how is the lung functioning, whether the drug is going to affect the lung functioning. All those studies are done in the respiratory system evaluation. So the next one is the effect of the drug on the GIT. So in these studies, it is analyzed that whether the drug molecule is going to affect the functioning of the GIT. That is, whether it is going to affect the motility of the GIT, or it may interfere in the secretions in the GIT, or it also helps to identify the GI related adverse effects. So all these things are done in the effect of the drug molecule on the gastrointestinal system. Renal and urinary system evaluation. So here it assesses the renal function, urine volume and electrolyte balance. Any abnormalities may indicate the kidney related risk factors are there in the drug molecule. Then S8, that is immunotoxicology studies. These tests are done to analyze the impact of the drug on immune system. The study is restricted to unintended immunosuppression or immunoenhancement response of the sample of the drug. See, these studies are done. What is the effect of the drug molecule on the immune system of the body? Whether it is going to suppress the immune system or whether it is going to enhance the immune system. So all these studies are done in S8 guidelines. Then S9, that is non-clinical evaluation for anti-cancer pharmaceutical products. So these guidelines are specially or exclusively only for anti-cancer drug molecules. So non-clinical evaluation typically involves a series of studies to assess the safety and efficacy before moving to the clinical trials. The guidance provides the information regarding those pharmaceutical drugs or anti-cancer drugs which are intended to treat the cancer in patients. Then finally, S10, photo safety evaluation. The photo safety assessment of a pharmaceutical product is an integrated process that can involve an evaluation of the photochemical characteristics, data from the non-clinical studies, and human safety information. So this test is done to analyze the photochemical characteristics of the drug molecule.